So good morning, everybody. Thanks for the invitation. I'm very pleased to be here. I have to say I've been impressed yesterday by the numerous dimensions that you cover here at IPSL and that were somehow illustrated by the different talks. You know, from nanoparticles to large-scale hydrology, climate, paleoclimate, environmental dimensions. And I guess today I open another dimension, also covered somehow at CEREGE, which is in the ocean, looking at biogeochemical cycles. So that's my specialty. I've been using numerical models for the last decades to study biogeochemical cycles in the ocean, with mostly two main questions in mind. One related to marine productivity by plankton, by phytoplankton, and how marine productivity responds to climate variability and climate change. And there are a few connections with teams here at CEREGE that also look at the interactions between climate and marine productivity, mostly in the past. And my second big question is about... Très près. OK. J'aurais une position un peu bizarre, mais... OK. And the second big theme of my research is linked to the carbon cycle and specifically to the capacity, the ability of the ocean to absorb carbon. And so maybe I start my presentation with those two main figures from the last IPCC report. You find them both in the summary for policymaker, in the synthesis reports, and I think they illustrate quite well the fact that we need biogeochemical models in the ocean to address some of the key questions we have to respond to today. So on the left here, you have a question related to the carbon cycle and what I will introduce a bit later, what we call the remaining carbon budgets, and on the right, a figure related to the response of marine ecosystems to climate change with potential links with adaptations policies. So I consider this figure on the left as probably the most important, the most policy-relevant figure of the last IPCC report. I guess most of you have seen it. It relates on the x-axis, the cumulative CO2 emissions from the start of the Industrial Revolution, and on the y-axis, global warming since pre-industrial. And despite, all the complexities of the climate system, it's quite astonishing to see that there is almost a linear relationship between those two quantities. And this is very powerful, policy relevant, because it tells you how much carbon we can emit in total from the pre-industrial if you want to limit global warming to a given threshold, to a given target. And this has been used a lot in the IPCC, in the political realm, during COPs for the Paris Agreement, to derive what we call the global carbon budget, or the remaining carbon budgets, for those different warming targets. So that's what you would find in the last IPCC reports. If we want to stabilize global warming below 1.5 degrees, starting in 2020 here, we have only 500 gigatons of CO2 remaining. We're already in 2025, so we've emitted more than 200 since then. So this remaining carbon budget is much lower than what's here on that figure. And on the second question, and you'll see that they are somehow related through the methodology and the tools. The second question, which is what's the impact of climate change on here fisheries catch potential or fisheries yield? This is the conclusion of the last IPCC report, saying that we expect a decrease in fisheries catch potential over the 21st century with medium confidence, and with high confidence, we project a larger decrease in the tropics. And you see here on the bottom right figure, a decrease of these fisheries yield of more than 25-30% at the end of the 21st century for a high emission scenario. So let me come back to this first figure, the one on the remaining carbon budget, and my argument here is that if you want to derive this very interesting relationship between global emissions and global warming, you need to use those kind of tools that I'm partly developing here. Yes, because from emissions to global warming, of course, you go through atmospheric CO2, and if you want to compute the trajectory of atmospheric CO2 from emissions, you need to know the future evolution of the land carbon sink, as discussed yesterday by Claire Cheney, partially, and of the ocean carbon sink. So it's only if you know how those things will respond to increasing emissions and to climate change that you can derive atmospheric CO2 and hence global warming from emissions. And what you see here at the bottom are projections of the ocean carbon sink in the coming decades as simulated by Earth system models, and I'll come back to those tools in a second, as simulated by Earth system models for different scenarios. And what you see here also is that for the different scenarios, from the high emission on the top here to the low emission scenarios in blue at the bottom, the uncertainties that you have from those bars, differences between models, is quite low. So the largest uncertainty comes from the scenario uncertainty, not from the model or the modeling uncertainty. And of course, as you see, if you want to be able to derive those relationships or those projections, we need or we use marine biogeochemical modeling, and I'll come back to that. For the second big question, which is the impact of climate change on marine ecosystems, I've showed the impact on fisheries yield on the top right here. But in fact, those projections, they are also derived from marine biogeochemical modeling. Why? Because in addition to global warming that you have here on the top left, you also need to know how much primary productivity the future ocean will be able to sustain, because this is at the base of almost all marine food webs in the ocean. And so those projections of marine ecosystem biomass in the next decades, they are derived from what we know about global warming projections in the ocean, but also about what we know on marine biomass of plankton projections in the coming decades. And again, for this specific question, the same type of tools are mobilized. So we use marine biogeochemistry modeling to derive the projections of plankton biomasses in the coming decades that then will feed upper trophic level models for example, to project fisheries or fisheries yield in the coming decades. So these are basically the two questions that we've addressed in the past decades, developing and using the tools I was mentioning at the moment. And this has been possible because the latest generation of climate models that Miriam described yesterday, mostly the physical parts, that's the one she described, they now include biogeochemical components in the ocean and over land. And for the ocean, we call them dynamical green ocean models, or we call them marine biogeochemical model. Somehow they've been developed for representing the ocean carbon cycle because of the ocean carbon sink question, but they also incorporate a simple description of the living biomass and specifically of the plankton. Why? Because the plankton is very important for the ocean carbon cycle. And so the fact that climate models do include those components has enabled projections of the ocean carbon sink, projections of ocean acidification, projections of deoxygenation that I won't talk about today, and projections of phytoplankton productivity in addition to all the physical components of the climate system. And so what you have on the right is a small animation from the IPSL model, so the model that we developed at the Institut Pierre-Simon-Laplace in Paris, in which we've used the physical basis of the model and we've incorporated biogeochemical components or biological components over land, and you have the leaf area index here on land, and marine biogeochemical components in the ocean, and you have surface chlorophyll in purple in the ocean. Just a simple seasonal cycle to show you how those models behave. And the best is, we are not the only one, of course. There have been many, many modeling groups, climate modeling groups at the scale of the world that have included marine biogeochemical components in their climate models. So at the international scale, there are about 40 different modeling groups that develop climate models. You know, those climate models that are then used for the IPCC assessments. So we have around 40 different climate models, and about half of them do include the marine biogeochemical components. And within this big international project, CEMIP, Couple Model Intercomparison Project, that is playing a large role, of course, in climate science in general, but in IPCC assessments in particular, we do run the same type of simulations, and we intercompare the output of those simulations quite a lot. We share the same database on this Earth System Grid Federation. It's a huge database that is shared by those different groups and that enable anyone from those modeling groups, but anyone outside of those modeling groups to use those data to do science and to look at what the projections and what all those simulations tell us about the climate in general. So let's come back to those two big questions, the evolution of the ocean carbon sink and the evolution of marine phytoplankton productivity. And I show you the latest results from CEMIP 6, so the sixth phase of the couple model intercomparison project. So those are the results that have been integrated in the last IPCC reports. And again, I'm showing the evolution of the RC carbon flux in the coming decades for the different emission scenario, from the high emission scenario here to a very low emission scenario in blue. And the line is, as always, the average of all the models that have been running the same simulation. And the envelope around the line is derived from the uncertainty from the differences between the different models. So we use the standard deviation of the different models here. And what you see is that for that specific question, the evolution of the ocean carbon sink, we have quite low model uncertainty. And so the IPCC states that we have high confidence in those projections. So there is much more agreement between ESMs in their ocean carbon response to the scenarios than it is the case for the land carbon response. So if you would see the same figure for the land carbon sink, you would have much more uncertainty around the mean projected evolution of the sink as compared to the ocean carbon sink. So why is it the case? Why do we have high confidence, little uncertainty for the ocean carbon sink? Because the main process controlling the ocean carbon sink, or the main processes, are physical and chemical processes that are represented very similarly in all the models. The ocean absorbs CO2 today and will absorb CO2 tomorrow, mostly because you have this difference in the pressure gradient of CO2 at the RC interface, and then carbon chemistry in the ocean, and then ocean circulation transporting this anthropogenic carbon to deeper layers. And because it's physics, because it's chemistry, because the models do represent with high confidence those processes, we have low uncertainty in those projections. So it's mostly the scenario that dictates how much carbon the ocean will absorb in the coming decades. It's very different if you turn to projections of net primary productivity. Again, same type of projections here. It's the relative change in NPP at the scale of the global ocean, compared between different scenarios. That's the different colors. And you have, in addition to that, the uncertainties coming from the differences between the different models. So the first point I'd like to make is about the mean projected trend. What we simulate is a decrease of marine productivity with climate change. A mean decrease. It's been the case for the past exercises, for the past IPCC reports. And this decrease of NPP in response to climate change is mostly driven by what we call ocean stratification. You know, with ocean warming, the ocean gets more stratified in the surface, and so you have less nutrient supply from the subsurface ocean to the surface, more nutrient limitation, less phytoplankton productivity. And this is something, a process, a driver that's been identified more than 25 years ago, and that's still the primary driver of those trends in those Earth system models. But what you see here is that around this mean trend, you see this decreasing NPP trend, you have a huge uncertainty, meaning that in some models, NPP does increase in response to global warming, and in some other models, NPP decreases much more than the model mean. And what happened even in the latest exercise in CEMIP 6 with the newest models is that this uncertainty has even increased as compared to what we had before. So we find an increase in net primary productivity, intermodal uncertainties in CEMIP 6, latest phase, as compared to CEMIP 5. So in terms of constraining the projections, the situation is even worse than what we had a few years ago, and I'll come back to this point in a moment. So you see that for those two questions that have somehow driven my career over the last 25 plus years, the type of questions we ask are quite different. On the ocean carbon sink side, we're more in a refining process. We're also exploring non-represented processes, especially on the biological side, because of the very low complexity of the models we use. So we still have new processes to explore. to be sure that the uncertainty we cover here is the right uncertainty, that we are not missing something important. On the right side, the question is quite different. We still need to understand the differences between the models, and at the same time, there are so many processes not represented in those models that we still need to explore some of those new processes. So this has driven most of the research we've been doing the last years. So maybe one word about the tool we use at IPSL, that is, by the fact, also used here at CEREGE. That's the tool that Yannick Donadieu and his team, for example, they use for looking at deep climate interactions between, variability in the climate system, the carbon cycle, deoxygenation in the Cretaceous, for example. They've been using the same tool. And so we collaborate with them for years now, for maybe even decades, on developing this model and on using these models in different contexts. So this marine biogeochemical component of the IPSL Earth system model is the PISCES model that's been developed for more than 25 years. You see here on the right, it's quite complex, but a simple schematic of what this model is including. And even if it seems complex, in fact, for representing the marine ecosystem, it's very simplistic. We have only 2 phytoplankton functional types, diatoms and nanophytoplankton. We have only 2 zooplankton with fixed parameters in time. So it's a very simplified representation of the plankton system. We do include, I would say, a more complex representation of many of the important biogeochemical cycles with 5 key nutrients: iron, phosphate, nitrates, ammonium, silicates, and carbon, oxygen, alkalinity, everything that you need to represent the carbon cycle. So this model has been coupled to many physical ocean models, so to the NEMO ocean model, mostly used for global studies. It's also coupled to some regional models like CROCO and ROMS for regional studies. It's included in 3 Earth system models at the international level, so in the IPSL model that I talk a lot about, but also in the other French Earth system model developed in Toulouse at the CNRS, and it's also the ocean carbon cycle component of the EC Earth model, which is another European Earth system model. And last but not least, PISCES is also the marine biogeochemical component of Mercator, which is, I guess most of you may know, the operational center for oceanography located in Toulouse that provides predictions of the ocean states for the next 2 weeks, almost every day, using PISCES for the biogeochemical components. So that said, what I want to do now in the remaining time of my presentation is to give you a few examples of current research, ongoing research, what we do now, exploring new processes, trying to constrain the uncertainty, and maybe also responding to new questions that I will briefly talk about. So all the results I will present now, they are all based on NEMO PISCES, using some ocean-only configurations or using the IPSL Earth system model for some of the questions. So this is the outline for the last part of the talk. A few highlights, as said, mostly ongoing work at IPSL, describing some of the work we do on better characterizing the ocean carbon sink, some work we do on better projecting ocean net primary production, and then I will say a few words about the fact that we can use those tools for new questions, and especially questions related to maybe the deployment of carbon dioxide removal techniques in the ocean, for which we have so much uncertainty still there. And I'll talk specifically about ocean afforestation for that last part. So the ocean carbon sink. So I have 2 pieces, 2 highlights. The first one is mostly physical, and the second one is mostly biological or biogeochemical. And they somehow concern very different timescales. But I think it's an illustration of the different types of work we do with those models. Okay, a few highlights. I'll talk about the skin layer. I don't know if you've heard about the skin layer. The skin layer is this micro layer at the surface of the ocean, just a few micrometers. That is very important because you have strong gradients of temperature, of salinity, especially in this skin layer. It's important, but it's not sampled correctly in the ocean. So when you measure PCO2 in the ocean, you don't measure PCO2 in the skin layer, you measure PCO2 in the bulk water, which is at a few meter depths. And in the models, it's the same. You know, you simulate the first layer of the ocean, it's 1 meter, so you simulate CO2 average over the first meter, so you don't represent what's happening exactly at the skin layer. And this may have consequences for projecting the ocean carbon sink, but also consequences for how we estimate the ocean carbon sink today. And this is what I will talk about now. So what you have on the left is a figure from the global carbon budget that Claire Chenu talked about yesterday. She mostly focused on the land side and the soil side. So this is the ocean component of the global carbon budget for the last decade. So we use, to estimate the ocean carbon sink in the past, in the last 50 years, we use 2 methods. We use global ocean biogeochemical models that I'm describing, but we also use database of PCO2 measurements in the ocean surface and then statistical methods to interpolate those measurements. And this issue with the skin layer that you see on the right somehow applies for both methods. In both methods, we miss the influence of the skin layer on our estimates of the ocean carbon sink. So what you see also is that those different methods using data products here in light blue or models in dark blue, they converge in estimating an increasing carbon sink, but there are still differences between those methods. So we are exploring mechanisms that could explain why those methods differ, especially in the past years in explaining, in estimating the ocean carbon sink. So we've included in our model a simple parameterization of the skin layer and how it may affect PCO2 and how it may affect the RC CO2 flux. And what we see in the model is that when you include the micro layer, this increases the simulated carbon sink by 5 to 15%. Why? Mostly because you take into account this micro layer, which is most of the time a cool skin with higher solubility of CO2 and then more uptake of CO2 in the surface ocean. So we've been playing with those new parametrizations to explore how that may affect our estimates of the ocean carbon sink. With the same type of models, we've been exploring new questions on the physics again, which is the impact of rain events on the absorption of CO2 by the ocean. And we've been looking at how rain affects the skin layer by reducing salinity that has an impact on CO2 solubility and on the RC CO2 flux. We've been also looking at wet deposition of carbon by rain events, and we've been re-estimating the ocean carbon sink using those new processes. So that's an example on how we collaborate with our colleagues that work on the ocean physics, that try to measure or parameterize the skin layer, and how we can then use those information to inform our modeling tools. Another highlight, very different, maybe more related to what many of you do here in CEREGE. We've been looking at what I call here the coral reef feedback for estimating the evolution of the ocean carbon sink in the coming decade. So that's something that is just missing from the models we use. The biological component of this ocean biogeochemical model does include phytoplankton, zooplankton, but we don't include benthic communities. We don't include coral reefs. And what we've been trying in the past year is to account for this potential feedback. The fact that with global warming, impact of global warming on coral reefs, we may expect reduced calcification. Maybe in some places we may expect net dissolution of the calcium carbonates. And we've looked at how this net dissolution or reduced calcification could impact the ocean carbon sink in the coming decades. So this is something that is well known for paleoceanographers. They've been working on those hypotheses for decades, since Brooker and many others in the 80s. But this hasn't been looked at for shorter timescales in the coming decades. So we've been using estimates of net carbonate production in the coming decades from specialists of coral reefs for the different scenarios. So this is net carbonate production for different emission scenarios in 2050, in 2100, and what you see is that in some of the scenarios, we expect before the end of the century, net dissolution. So this would change the balance of alkalinity in the ocean and hence the ability of the ocean to take up CO2. So we've been playing with the model, introducing this as a new parameterization, so addition of carbon and alkalinity, taking into account the dissolution of reefs, as a function of today's reef distribution and as a function of the different scenarios. And when you do that, I mean, intuitively for us, sometimes it's counterintuitive for some others, but of course, if you dissolve coral reefs, you will increase the ability of the ocean to take up CO2 because of the addition of alkalinity in the ocean. And that's what we simulate here. So that's at the end of the century, the change in alkalinity, so addition of alkalinity in the ocean. And in terms of the ocean carbon sink, it's not a primary effect, but it modifies, you see here in 2100, for all the scenarios. So this is a high emission scenario, expecting net dissolution of coral reefs, and you increase the ocean carbon sink if you take into account these effects. And this would have consequences on what I told you about before, the remaining carbon budget. We have to take into account all those feedbacks if we want to reduce the uncertainty or if we want to be more accurate in estimating those estimates. So the reef dissolution induces an increased ocean sink up to 15% in 2100 for one of the scenarios, and this is about the same magnitude that the differences between the models that I was talking about before. So it's an effect that we have to include in the models in the coming years. Second part with, again, two highlights, looking at ocean net NPP. And the first one is specifically addressing one key process that is represented in models, but in a very, very simplistic way, which is biogenic N fixation by some organisms, mostly cyanobacteria in the ocean. And we have some key unknown on how N fixation may respond in the coming decades, and I will show you that it may have a first-order effect on our projections of NPP, which was kind of a surprise for us. So coming back to IPSL, here I'm contrasting the projections of two versions of our IPSL Earth system model. On the left here, you have IPSL CM5, which is the version that we used for the previous CEMIP exercise, the previous IPCC report, and you have the new version that was used in the last CEMIP exercise. So even if it's different scenarios, but similar in terms of emissions, you see that the warming signal of those two climate models is quite similar, with North Pacific warming more than elsewhere, same in the North Atlantic and in the Southern Ocean. But the response of NPP, global NPP, in those two models differ tremendously. In IPSL CM5, in black, we were simulating a decrease of NPP at the global scale. In the new model version, we simulate an increase of NPP at the global scale. That's quite surprising, but also a bit frightening. When we use those projections then to feed ecosystem models and look at fisheries in the coming decades, if we don't even know the first order signal in terms of how NPP responds to climate change. When you look at the regional projections of NPP, so these at the bottom are the changes in NPP in CM5, in CM6. You see some common patterns, but you see specifically in the oligotrophic gyres, in the subtropical gyres, you see decreasing NPP in the previous version, and you see now increasing NPP in the new version. What has changed? We're still using the same model, PISCES, with some new developments that have been included. We're still using the same components of the climate models, so something fundamental has changed. And what has changed is the way we parameterize N fixation. This process that to us seemed to be second order in many instances, for the global NPP seems to be first order. And what you see here is that in the new version, we have N fixation increasing tremendously across the 21st century, responding to global warming. Basically, we have more and more N fixers in the ocean. So you have an input of new nitrogen that not only sustain productivity in the oligotrophic gyres against stratification, but because of this additional nitrogen input, you have even more NPP at the end of the century. And this response of nitrogen fixation is key, but it's non-trivial. It's quite subtle. It depends on how you parameterize the stoichiometric ratios of N and P in your organic matter. It also depends on how you parameterize the response to temperature of N fixation. And so we need to do much more on that specific process for those specific regions because it seems to control quite a lot the response of NPP at the global scale. Okay, last point on on the biological part, just to tell you about some new work we're conducting. So I was telling you that the kind of model we use are very simplistic. You know, a few phytoplankton types, a few zooplankton types. But specifically, we use fixed parameters for the physiology of these organisms. So they don't adapt, and they don't evolve. And that's true for most of the traits, except for temperature. For temperature, we assume that they adapt perfectly to temperature, with increasing growth rates, with increasing temperature. It's like a perfect Darwinian model for temperature. And what we've done with a colleague recently is to look at the potential influence of adaptation, of evolution of those organisms in response to climate change, to warming in particular. And so to do so, you have to modify your model tremendously. So what he did is that instead of having just one phytoplankton type with some fixed response to temperature, like increasing growth rate to temperature, like an Epley curve for the ones that know the work of Epley here. So we've been transforming this one phytoplankton type or one zooplankton type here by 40 different classes, all adapted to different thermal niches. And then we can use those new representation and include some representation of evolution processes with a modification of the parameters along time. And so we're playing with this, looking at how including or not adaptation change our assumptions, our results. And what you have here is an effect of maladaptation of mesozooplankton. So if you assume that there is no adaptation at all, as compared to perfect adaptation of mesozooplankton, you would reduce biomass considerably in the ocean. So that's the type of questions that we focus on, working a lot with our colleagues, biologists working on evolution of phytoplankton and zooplankton. Okay, last small part of my presentation. I've been mostly talking about projections in response to anthropogenic emissions. I want to tell you that we are also using those tools now to explore some new questions, specifically questions related to carbon dioxide removal techniques in the ocean. So I assume most of you have heard about iron fertilization. It's been discussed for more than 30 years. So we've been exploring the efficacy, the efficiency of iron fertilization using those tools. But I'm showing now some new results on another MCDR technique that has been explored only for the past few years, which is ocean afforestation. And I will show you some of the recent results we obtained. So what is ocean afforestation? It's the aim of using macroalgae in the open ocean to capture CO2 in the organic matter, and then to have this organic matter transferred to the deep ocean, where the carbon, after remineralization, may be stored for decades to centuries. And it's kind of a weird question, maybe weird method to explicitly cultivate macroalgae in the open ocean, but it's based on the fact that we do cultivate macroalgae on the coast for millennia, especially in East Asia. It's based on the fact that we have 2/3 of the planet that is covered by ocean water, and so the competition for space, of course, in the ocean is not the same than the one we have on land for bioenergy, for example. And so we've played with the model, introducing a simple representation of macroalgal cultivation. And what we do with the PISCAS model is, of course, when you cultivate macroalgae, you consume CO2, you consume DIC, but you also consume nutrients, nitrate, phosphate, iron. And so you will feed back on the phytoplankton if you consume nutrients. Because the assumption here is that once the organic matter is produced, it's transferred to the deep ocean to store the CO2 there. So with running simulations in which you cultivate macroalgae in the different exclusive economic zones of the world, and we've looked at what's the effectiveness of this technique, we've looked at what's the impact on phytoplankton that macroalgae cultivation in the open ocean or in the coastal ocean would have. And what we show is, when you do that, the first result is that it's not because you absorb 1 mole of carbon in macroalgae that you transfer to the deep ocean, that this mole of carbon will be replaced by 1 mole of carbon that would come from the atmosphere. This seems quite intuitive, but most of the time when, many of the startups, they just estimate the potential of their MCDR techniques, they relate a mole of carbon captured by their technique to a mole of carbon that would be removed from the atmosphere. Why is it the case? Because, of course, then it depends on the chemical property of the ocean surface for that CO2 molecule to be replaced by the CO2 molecule that would come from the atmosphere. So we've looked at this efficiency, and what you find is that it's only 70% or 75%. And then when you take into account the feedback on phytoplankton, it's even worse. Because when you do cultivate macroalgae, you consume nutrients that you will store in the deep ocean, and you will reduce nutrient availability for phytoplankton, and so you will reduce the efficiency of the biological carbon pump. And then you have an efficiency of that technique as compared to the total macroalgal carbon production that is even less than 50%. And what we show also is that when you cultivate macroalgae in the EEZs, here on the coastal ocean, most of the flux would occur outside of the EEZ. So that also is a huge problem for what Claire Cheney was talking about yesterday, monitoring, reporting, verification. How do you monitor? How do you verify that the carbon is indeed coming from the atmosphere? So that's just an example, but I think it's a nice example to show how we can use tools that were not designed for those questions, for new questions related to MCDR. So limited effectiveness due to the residence time of surface water, to RC equilibrium, to competition with phytoplankton, localized uptake outside EEZs, and also substantial impact on phytoplankton production. Maybe time to conclude. So maybe I come back on those two questions. I talked about high confidence. We still need to reduce uncertainties for these ocean carbon sink, especially in the case of mitigation scenario and zero emission commitments. And also we're working hard to reduce uncertainty because, again, if you come back to MCDR, what we'll need is a very clear ocean baseline if you want to be able to monitor and verify and report CDR efficiency. And then on the NPP, you've seen that we're still working hard, exploring new processes, trying to develop methods to reduce uncertainties, and trying to use those projections, even with these uncertainties, for informing conservation adaptation strategies. Hard, but there is no choice about trying to do that. And that's the end. Thanks a lot.
Thank you. We have time for a couple of questions.
Fantastic talk, thank you so much. My question is regarding the changes in NPP over sort of anthropogenic timescales, where you show really nicely the importance of stratification, particularly. I guess my question is it reasonable to treat the ocean as a closed system on those timescales? We know we've accelerated both nitrogen and phosphorus input to the oceans significantly in that time. So how do you factor in that to the model simulations?
Yes. So in fact, we do include changes in. We do include changes in atmospheric deposition of nitrogen and phosphorus in those models. So we do use projections run by atmospheric chemistry model, in which they take into account the change of the global nitrogen cycle. So we have nitrogen deposition scenarios that do affect the response that I'm showing here. Not in all models, but in many models. So we've explored those, both from rivers and atmospheric deposition. It's important locally, maybe regionally, but at the global scale, it seems to play only a second order effect, yes.
Thanks. My question relates to paleoclimate. I was wondering whether, in your opinion, there are any paleo targets related to ocean PP that could actually help reduce the uncertainty in NPP model projections, or does the qualitative nature of export productivity records in the past mean that it's essentially useless in helping to inform models?
Yes. Maybe two elements to respond to your question. The first one is that NPP is not export. And when you look at the projections, they do diverge tremendously in terms of NPP, but when you look at exports, they are much more convergent. So in fact, you have a decoupling between, with this N fixation story, you have a decoupling between NPP and exports. In this specific model simulation, I have decreasing exports, but increasing NPP. So, of course, using the paleo record would mostly constrain export projections, because that's, I guess, mostly what we record. And yes, we've tried that. I have a few examples in mind, maybe just one. We've looked with the same model tools at how NPP or exports would respond to Heinrich events, for example, in the past. And the type of timescales is interesting, I guess, maybe relevant for what we're looking here. And we've looked at the North Atlantic specifically, where we have maybe sufficient records to demonstrate a decreasing exports in response to some Heinrich events. And with the model, qualitatively at least, we were reproducing that response. So that would help us constraining the response maybe of exports, which is much easier to project than NPP.
Thank you, Laurent, for this very nice presentation. I was very interested in your surface microlayer results, and I was wondering, I was very impressed by the effect of taking it into account, and I was wondering if you take into account the content of organics in this microlayer, and if it could affect the CO2.
No, we don't. Here it's really only the physical processes through changes in temperature and salinity. And we haven't looked at organics. But yes, there have been, for CO2, I don't know, but for many other trace gas emissions by the ocean, it's probably the most important factor when simulating isoprene emissions or some other type of emissions, and we don't do that well. So that would be a next step for us. Yeah.
Okay, thank you.
Thank you for your talk. I could have many questions, but I will focus on one, which is about dissolution of reefs. In this study, did you estimate the omega aragonite, and how much aragonite do you dissolve? How many years of a reef do you dissolve? What is the thickness of? Can you control that?
No, in fact, what we've done in the model is very simple. We've used the estimations by Cornwall. So we've used directly their net dissolution estimates. And to do so, they've used mostly the effect of warming and of omega or acidification. But we haven't explicitly represented the processes of dissolution in the model. In the model, we only add DIC and alkalinity. That's the only thing we do. So in the next step, I have a postdoc working on representing specifically reefs in the model, and then we'll be able to do that. What we see in Cornwall is that, of course, there are huge uncertainties on today's calcification. I don't know, maybe a factor of 5 at least, 30 maybe to 300 teragram of carbon. Huge uncertainty. There's also a huge uncertainty on the stock of calcium carbonate that you may be able to dissolve. I'm not a specialist. From what I've seen, we have more than enough for the type of scenarios we've explored here.
Thank you very much. My question is about the boundary layer that you are calling the skin layer. But actually, I guess it's more than the skin layer. It's also the atmospheric layer, which is right above the liquid water. And my question is, do we have enough data on the climate or even the physics on this little atmospheric layer above the water layer, or should we need to focus more on this special area?
I think for
constrain the model.
I think for the CO2, it's mostly controlled by the water boundary layer. So on the water side than on the atmospheric side. For some other gases, it may be different. And yes, we have lots of difficulties measuring what's happening there because of the special scales that are so little. So I have, on the water side, I have colleagues developing tools to extract the first micrometers to measure PCO2, to measure DIC and alkalinity, temperature, salinity, of course. But on the atmospheric side, I don't know, I have to say. Yeah.
Maybe we stop the discussion now so we can continue with the other speakers. And yeah, I mean, you can talk with Laurent later.